Hello, everyone. Happy Thursday, and welcome back to Science in the Age of COVID-19 seminar series. The coronavirus pandemic has been raging for quite some time now, and there are many interesting aspects of this virus infection that we are observing. First of all, there appears to be a range in severity. On the one hand, we've had record number of lethality, while on the other hand, we have asymptomatic carriers walking around among us. There also appears to be a difference in susceptibility, especially with respect to sex and age. Men and the elderly appear to be more susceptible to the virus infection. Another interesting aspect is cellular response, immune response at the individual level. While a strong, robust immune response is critical to fight off any infection, a hyperactive immune response, or what we call a cytokine storm, appears to be the main causative agent of lethality with respect to COVID-19. And our speaker today is going to shed light on some of these differences, some of these aspects of the virus infection. So it gives us immense pleasure to welcome Dr. Angela Rasmussen to our seminar series today. Dr. Rasmussen is an associate research scientist at the Center for Infection and Immunity at Columbia Mailman School of Public Health. She employs a combination of classical virology, genetics, and modern systems biology techniques to understand some fundamental principles governing host pathogen response. And today, she's here to share with us some very recent findings about SARS-CoV-2. So without taking up further time, Angela, I virtually hand you over to speak.
Well, thank you so much, Sarada, for having me here, for inviting me to join this seminar series. I'm really looking forward to telling you about some of my research, some of the other research that's been going on in the field with regards to the host response to SARS coronavirus 2, and most importantly, to talk about the things that are still unknown and that we still need to look at. So that's going to be the subject of my talk today. It's mostly going to be about MERS coronavirus and SARS coronavirus 2, but I am going to manage to toss in a little bit about Ebola as well, since that is my original true love in terms of virology. So let me advance my slides. Okay, so before I get started, I'm going to do a territorial acknowledgement as well as an equity statement. And this is really important, and I've begun doing this for all of my talks to just let you know that today I'm in Lake Bay, Washington, presenting from the unceded ancestral homelands of the Coast Salish people, and specifically the Puyallup tribe. Much of this analytical work that I've done, it was performed either on the traditional lands of the Duwamish people in Seattle and the Lenape people in New York City, Manhattan. I acknowledge and honor the first people of these territories, as well as their tribal governments and their histories and ancestry and their roles today in caring for these lands. And I'd also like to acknowledge that there is a history of systemic inequity in academic science that spans decades and really centuries. Columbia University itself, my institution, was founded using profits from the transatlantic slave trade, and that's left a long and painful legacy of racial and gender-based inequities that continues to this day in academic research and medicine. And I encourage everybody here to consider how you can contribute to making scientific research a more equitable enterprise. And with that, I'm going to begin my presentation. First, I'm going to define what we think of when we think about systems biology. So I suspect that if you talk to anybody who considers themselves a systems biologist and ask them what is systems biology, you're going to get a different answer. So I thought I'd start off by telling you how I think about this. And I really think of it as this iterative cycle of different research and analytical activities. So I like to start with an experimental model system, and that can be cell culture, it can be an animal model, it can be samples from human subjects or patients, and doing the really basic virology, biochemistry, the type of bench science that many of us think of when we think of doing virology research. I take samples from these different models and I use them to generate large omics data sets. Primarily, I work with transcriptomic data, so a global survey of the host gene expression landscape. I also do some work with proteomic and metabolomic methods to look at how proteins and metabolites are regulated in response to viral infection. Then you take all of that big data and you put it together with bioinformaticians and computational biologists who will use different models to make sense of all of that data and try to filter out the grains of truth from the noise in these large, very multidimensional data sets. From there, you can find these keys really to the understanding the biology of the system and apply them to different translational applications. So biomarker panels that could potentially predict disease outcome or be used for diagnosis, novel drug targets and targets for drug repurposing, which I'm going to speak about a little bit, correlates of protection from either naturally derived immunity or a vaccine. And then you can really repeat the cycle. So you can use this to generate hypotheses and then test them using similar approaches. And I found that the combination of multiple types of experimental systems and analytical models can really be very effective at answering complex questions, including those that involve the host response. So the host response is really one side of a coin if you think about pathogenesis. And there's really, there's two components to any pathogenesis of any type of viral disease. On one hand, we have the virus. In this case, I'm showing the genome of SARS coronavirus, or SARS classic, from 2003. Coronaviruses, as most people these days know, have a relatively large genome for an RNA virus, but it's still pretty small compared to the host. It's 30 kb, encoding 26 genes that encode multifunctional proteins. And though there are, many functions of these proteins are very pleiotropic, it still requires the host in order to replicate, just like every virus. And every virus also is specific to the virus family that it's in. So there are many viruses that do common functions, but they often encode them in different ways across families. So there's a lot of diversity in the world of viruses. When we think about the host, it is actually one thing that human pathogens have in common. There's just one host for human pathogens, at least, well, not always, but that we're interested in anyways, and that's us. And our genome is much larger than all viral genomes. It's 3.2 megabases, 3,200 megabases, 3.2 billion base pairs. Those roughly encode 20,000 coding genes that not only encode multifunctional proteins, but different isoforms of those proteins that are organized into different specialized pathways. So there's really two scales present here. While the viruses are smaller and I wouldn't say simpler, but I guess comparatively in terms of size they are, the human response is something that all viral infections have in common when we're thinking about human pathogens. And so I focused on that in my research, just because we don't necessarily know which virus is going to emerge next, but we know that we are interested in all of those viruses having the same host. And we do know also from a great deal of work, this is just two examples, that host responses to infection really do drive pathogenicity. So on the left here, we have an example of two different influenza virus infections. These are in cynomolgus macaques. On the left heat map, we have Texas 91, which is a seasonal flu strain that's less pathogenic. On the right heat map, we have H5N1, a Vietnam 1203 strain, which is highly pathogenic, high mortality rate. And you can see that these host responses transcriptionally are very different. The seasonal flu has a much more muted response. Some of these pro-inflammatory genes or cytokines and chemokines are actually downregulated early in infection, whereas in H5N1, they are lit up across the board, showing that there is this massive unregulated inflammatory response occurring to infection. On the right example, we have Ebola virus and Reston virus, and this is an infection done in human monocyte-derived macrophages. Pardon me. And we can compare Ebola virus, which is highly pathogenic, again, very high mortality rate. Most of us know about Ebola, compared to Reston virus, which isn't really pathogenic in humans at all. And you can see again that they are very different host responses, even in a cell culture model where you can't actually look at pathogenesis. So this really shows that across different viral families, the host response is really important for determining whether a virus is really just a nuisance or an incidental infection, and whether it is actually a severe pathogen that presents a major public health threat. So one thing I've been thinking about is, can we use this information to prepare for viruses that we don't know about before they emerge? And that has got me working on some of my older data about MERS coronavirus, trying to think of new ways that we can leverage what we understand about the host response to coronaviruses to apply to SARS coronavirus 2, as well as any other viruses that might emerge. So one theme of my work has been trying to understand this concept of tolerance. So tolerance is the idea that you might get infected with a virus, but who cares, because you're not going to get sick from it. And this really has implications that are translational. So the status quo of kind of how we develop drugs or countermeasures to virus infections is to get rid of the pathogen, to develop antiviral drugs that will reduce pathogen load and presumably help the patient recover. And what we've seen in some of the more recently emergent highly pathogenic viruses like Ebola, SARS coronavirus 2, is that antivirals have limited efficacy, in the sense that when patients are very ill already, it's not necessarily the virus anymore that's completely driving their clinical symptoms. And so getting rid of the virus at that point, when the patient already has this harmful host response occurring, may not be very helpful to them clinically. So normally the way that antiviral development is approached is that you identify the pathogen that you're trying to target. You learn about how it infects cells and causes disease. You can exploit that information to develop a novel drug that disrupts one of those processes. And then you develop that drug, you target that mechanism, you learn how that drug works. You test it in animals, you test it in human clinical trials, you get FDA approval, you market it, and then you can address emerging drug resistance that can occur against that antiviral drug. If you instead are looking to tolerate the pathogen, you don't even really worry as much about the pathogen at all. You can leverage the existing data that's out there about host responses to many different kinds of pathogens across potentially multiple viral families, multiple pathogens that cause multiple types of disease or type of disease in common, such as hemorrhagic fever or respiratory disease. You can identify those host responses that are really driving those different disease states, correlate those with outcome, identify key host regulators and target those, and you may be able to target those with a drug that already exists, depending on what those are. And then you can filter those candidates by testing them in vivo in different experimental animal models. And then theoretically, you could rapidly deploy these. And this would be a much faster path to being able to respond to an emerging pathogen that we've never seen before. And again, in this scenario, we don't really care about the pathogen load. We can let the body take care of that. The immune system can clear the virus. But by modulating the host response, we're actually ameliorating some of the more severe clinical symptoms. And that theoretically would allow the patient to recover and fight off the virus on their own. So a lot of what I'm going to talk about today is trying some of the work that we've done to understand the host response to coronaviruses and to SARS coronavirus 2, and how we might apply that information, both for SARS coronavirus 2 as well as other pathogenic coronaviruses that might emerge in the future, because there are certainly many more that might have that potential. So as I mentioned, I'm going to talk a lot about some of these differential models of MERS coronavirus disease that we've been developing in collaboration with Vincent Munster at Rocky Mountain Labs and Dick Bowen at Colorado State University. And then I've been also working with Ben Garcia, who has done a lot of global proteomics and metabolomics to complement the transcriptomics studies that I've been leading. So what we've used 4 different animal models in the HDPP4 transgenic mouse model, which is a transgenic mouse that expresses the human receptor for MERS coronavirus DPP4. There is a dose-dependent tolerant effect. So if you dose the animals with a high dose of virus intranasally or intratracheally, the animals will die. All of them will die. If you treat them with a lower dose, they will, some of them will still die, but some of them will be able to survive infection. And so we've been comparing host responses between those two different dosing groups to look at what are determinants of survival. We've also been comparing across species, both in natural models of tolerance, such as the dromedary camel, which is one of the natural reservoirs for MERS coronavirus. Dromedary camels are not completely tolerant in that they do have some very mild clinical symptoms. They have increased mucus production, and camels are generally bad-tempered anyways, but apparently they're worse tempered when they are infected with MERS. And so they are a really natural model of mostly tolerant, very mild disease. The alpaca is a completely tolerant model in that they are infected with MERS productively. They do have increased virus loads and virus shedding, but there are absolutely no detectable clinical symptoms associated with infection. And we compare those to the marmoset, which is a very severe, probably lethal. We euthanize the animals prior to them meeting clinical endpoint criteria for humane euthanasia or being allowed to die naturally. We don't let them get that far, but they do develop very, very severe pneumonia. And so we are able to contrast across these species to look for sort of universal determinants of tolerance or lethality. And right now I'm going to focus on some of the data that we have generated looking at signatures of tolerance in the mouse model. And so in this experiment, we had 3 groups of mice, time-matched mock-infected animals, 6 mice per group, as well as a low dose of MERS coronavirus, which is the tolerant condition, and a high dose condition. And then we looked at host responses to infection by transcriptomics, proteomics, and metabolomics. And today I'm going to be focusing on the transcriptomic data since that is what I know best. Again, in these mice, they are all infected productively. However, the high dose group is uniformly lethal. The lower dose group is not. We also see that in all of these animals, there is both viral RNA and total RNA of the virus, as well as messenger RNA, subgenomic RNAs, indicating that the virus is in fact replicating. And these experiments were done in the BSL-4 lab at Rocky Mountain Labs. So when we looked at host responses in the lungs of these animals, we saw that the tolerant animals at the low dose had a much more tightly controlled host response compared to the lethal group. And even though there is some overlap, which you would expect, because even in the tolerant group, there is still pathogenicity. And that's what we can see here in this multidimensional scaling plot. This is a way to take high dimensional data, such as transcriptomic data, where you're looking at thousands of different genes, and compress that information into a 2-dimensional visualization. So the difference between these samples is given by Euclidean distance. So the further away a dot is from another dot on this plot, the more different they are. And we can see here that the high dose animals do have a unique host response compared to the low dose, which at early time points is actually very similar to the mock infected controls. And we can see this too when we look at functional enrichment. So we used Ingenuity Pathway Analysis, which is a functional analysis software that looks at a large manually curated knowledge base of different interactions and biological functions as reported in the peer-reviewed literature for all human, mouse, and rat studies. And what we see here is that while both tolerant and lethal animals do mount an antiviral response in terms of interferon signaling, the response in the tolerant animals is much more muted at early time points, especially. It's not really until day 5 that these animals begin to mount a more robust inflammatory and immune response. And that's likely because at this point, they are beginning to control the infection in the animals that survive. They are regulating their host response to the infection rather than just coming out sort of with all guns blazing, which is what happens in the animals that have lethal disease. They have early upregulation of a lot of these different inflammatory pathways, followed by sort of a downregulation of the response, suggesting that coming out sort of with, again, all guns blazing, this strong, unregulated inflammatory response allows the response to essentially become non-functional as the infection progresses. So these are pretty different host responses that we're observing in both the tolerant and the lethal condition. And we were definitely curious if this would be the same across other species. And we do see when we look at, we compare the alpaca model, which again is a complete model of tolerance, to the marmoset model in which the animals have lethal disease. We see a similarly muted host response in the tolerant alpacas. Effectively at day 3 post-infection, again, these are in lungs of animals that were serially euthanized. We don't really see any kind of functional enrichment at day 3, and we see that come up later at day 6. In the marmoset model, a lot of these antiviral pathways are actually downregulated, but in lesions from these animals' lungs, we see that there is a lot of inflammatory signaling, again, suggesting that disease, and in this case, actual pathology in the lungs, in these lesional sites that we looked at specifically, is really occurring in an out of control way. And here we actually looked in the marmoset at 2 different MERS coronaviruses. One is the Erasmus Medical Center isolate, that's sort of the prototypic MERS coronavirus strain. And the SK6 is a strain that was isolated from an outbreak in South Korea in a hospital. So I wanted to compare this to some of the limited data that we have available about host responses to SARS coronavirus 2. And from what I've seen in the literature, there's only really 2 studies that have looked at this in the context of either patients or animal models. And so one study that came out of Ben Tenover's group at Mount Sinai was just published in Cell, and it found that both in COVID-19 patients, as well as a ferret model, which is a mild model of SARS coronavirus 2 or COVID-19, as well as in normal primary bronchial epithelial cells, the infection is characterized by a reduced antiviral response and that leads to an upregulated inflammatory response. So that's very similar to what we've seen in the MERS coronavirus model, where antiviral responses occur. They may be somewhat blunted by infection, but it also is associated with this increase in inflammatory cytokine production and inflammatory mediators, inflammatory signaling. So this is really consistent with what we've seen in MERS. And similar things were found in this paper by Chris Mason's group at Weill Cornell, looking at a number of specimens from COVID-19 patients. This is, I think, really independent of outcome. These are clinical specimens that they had, so this is really only associated with infection. But again, we do see upregulation of many of these different inflammatory mediators associated with COVID-19 disease. Presumably, many of these patients were actually hospitalized because that's how the samples were acquired. So we might assume that they had more severe disease, but we definitely need to do more work to try to distinguish host response signatures in patients that have mild disease or might be more of a tolerant COVID-19 phenotype versus patients that end up with this very severe either pneumonia or one of the many other diverse presentations of COVID-19 that have been observed so far. So we also are continuing to work with our collaborators at RML, Vincent Munster and Emmy DeWitt, to look at host responses in a rhesus macaque model. And so they recently published on this model. They infected rhesus macaques with SARS coronavirus 2, the Washington 1 isolate. And they see in this model that, in the first 10 days, roughly 7 to 10 days post-infection, the animals do have observable clinical disease. They have virus detectable in nose swabs, in throat swabs, in bronchoalveolar lavage fluids, both RNA as well as infectious virus. And they do have pathological signs of interstitial pneumonia. So they not only have observable pathology at the time of necropsy in terms of just gross pathology that you can see with the naked eye, you can also see infiltrating inflammatory immune cells, and you can also observe signs of pulmonary infiltration, pulmonary infiltrates consistent with human disease when you do an X-ray of these animals. So from the study, we collected whole blood samples over a 21-day time course, and we've done RNA sequencing to look at systemic host response signatures. And unfortunately, I wish I could share this data with you, but it just came off the sequencer on Monday, and so we haven't gotten to the point yet where I've been able to do a functional analysis. So sadly, if this had been 1 week later, I probably would have had preliminary functional data to share with you. But for now, just stay tuned, and I'm going to get out a preprint of our analysis as soon as I possibly can. So now I'm going to talk to you about one of the applications of this type of data collection, and that's using machine learning techniques to predict the clinical outcome of these different viral infections. So I'm going to talk about this a little bit later in the talk, but we've actually used this pretty successfully with a mouse model of Ebola virus disease that we've developed. This is a genetically diverse mouse model in which some mice of some genetic backgrounds are tolerant to Ebola infection, meaning they lose weight, have very, very mild disease, and fully recover, compared to the other 2/3 of the lines in this model, which are either uniformly lethal or mostly lethal. And about half of those, a third of the overall animals, develop hemorrhagic disease as well, where there's evidence of disseminated intravascular coagulation, prolonged clotting times, other signs of coagulopathy that are consistent with what we see in some Ebola virus disease patients. So we decided to apply that same method that we've used to try to predict outcome as well, using first these MERS coronavirus models, and then we hope to apply this to SARS coronavirus 2. And how this works is we take these host gene expression profiles, we look at genes that are either highly upregulated or downregulated. So things that would be detectable with a PCR assay that would be in really most any clinical laboratory around the world. And we look at each of those and their ability to predict this, and we iteratively filter out genes that meet this expression criteria that are not actually very good predictors. And then we use that to train a model called random forests, which allows us to classify these genes, and we see how well it works. And we train this model using 60% of the data chosen at random, and then we test it on the remaining 40% of the data. And we do this 1,000 times to make sure that we are accurately estimating our predictive performance. So in the case of the MERS coronavirus mice, we identified a signature of 7 genes that are either highly up or down regulated in the uniformly lethal group, not so much in the tolerant group. And when we ran our model and looked at predictive performance, we were able to predict outcome correctly 90% of the time, roughly. So this is a good sign, certainly, that we could use this technique to predict outcome across other models and potentially, eventually, in patients. So I just wanted to give you an example of how well this worked for Ebola. As I mentioned, we were using the collaborative cross, which is a genetically diverse mouse model with differential outcomes for Ebola. And in that model, using just the mouse data, we were able to predict outcome with incredibly high accuracy. So this is almost 100%. It's 99.5% predictive performance. And then we applied this to a data set from human Ebola virus patients collected in Guinea during the West African Ebola epidemic. And the group that had originally sequenced these samples also applied some of these techniques and were able to predict outcome with 79 to 85% accuracy, which outperformed the sort of standard way of trying to predict outcome, which is usually based on CT value at the time of diagnosis. So the lower the CT value, the more virus you have and the more likely you are to die from Ebola virus disease. Their predictors developed with the human data outperformed this and were about around 80% accurate, depending on the model that they used. So we took our mouse-derived classifier, we mapped it to the human orthologs, and we tested it with the human data set. And in that circumstance, we were able to predict outcome correctly in 75% of the tests that we ran. So that's good. It's right around the same range that they saw, and it's actually better than CT value as well. It's probably not good enough for a clinical assay. So we thought about ways that we could actually improve this, and we added data from a non-human primate model that we've been working on with Tom Geisbert at UTMB. And once the NHP data was included in this, we were able to predict outcome correctly 95.6% of the time. So the more data that we have across more species, the more accurate our predictions become, and hopefully the more applicable they would be to human patients. So we really hope to do the same thing, not just with SARS coronavirus 2, but to also leverage the wealth of data that we've generated for MERS coronavirus, as well as the wealth of data that exists in the public domain for SARS Classic, for MERS, and now increasingly for SARS coronavirus 2. So that's coming up next. And really one thing that we'd like to do going forward is to start predicting disease features across viruses. So who cares if it's a coronavirus or if it's a new influenza virus or if it's some other virus that's causing respiratory disease that we weren't prepared for and we didn't expect? Can we identify signatures that would predict outcome in these cases? And so this is a study that I've been working on with Michael Kirby at Colorado State University. He's a mathematician who has developed his own custom machine learning algorithms to do this type of classification. And this was using microarray data that was collected from human subjects that were experimentally infected with low pathogenicity seasonal influenza strains, respiratory syncytial virus, and human rhinovirus. And this was trying to see if we could classify the patients that would shed virus before they actually had detectable virus being shed. So we were actually able, across all of these different virus families, across these different experiments and cohorts, able to cleanly separate these different shedders from the controls. So this bodes well for our ability to apply these types of approaches to classifying outcome, even when you don't know which virus is causing the actual disease, which would make this very useful for future emerging viruses, especially those with epidemic or pandemic potential. So this is just to summarize how we're going to approach this. We're going to, as I mentioned, take our transcriptomic data from the MERS coronavirus animal models, extend this to SARS coronavirus 2 animal models, and hopefully some patient data, and refine this further with all the data that's in the public domain from all different pathogenic beta coronaviruses. And so hopefully this will be applicable to other virus families as well in the future. And now I'm going to talk about another type of translational application for this type of host response analysis, and that's really mining this data for druggable targets and repurposing candidates. And the idea here is that if you find pathways that are associated with tolerance, we can assume that perhaps activating those pathways in any patient might lead to a tolerant outcome. And conversely, if we can find pathways that are associated with highly pathogenic or negative clinical outcomes, we could inhibit those pathways and also hopefully improve patient outcome. And this can be applicable to either drug repurposing, which is essentially finding existing drugs that target the molecules that we would discover using this type of approach, and see if those can be rapidly deployed to modulate these host responses. Also, I'm not going to talk about it today, but you can identify upstream regulators that regulate these pathways that are either protective or pathogenic, and potentially develop new ways to target those molecules upstream. So here is the drug repurposing work we've done for MERS coronavirus. This is actually a combination of proteomic, transcriptomic, metabolomic data. We selected for the most predicted drugs that are, first of all, available from the FDA. They are FDA approved and available for prescription. And we prioritize those that are the ones that both inhibit lethal gene signatures and then activate tolerant gene signatures. Although we also are considering drugs that would be predicted to do one or the other. And I just thought that I would point out a couple things here. And one is that we identified dexamethasone, which as of yesterday was reported, at least in a press release, to be effective at treating patients with severe COVID, probably by the mechanism that allowed it to be identified here. And that's that dexamethasone is a potent anti-inflammatory that would presumably dampen those harmful inflammatory cytokine storms that have been associated with severe COVID, and potentially activate signatures that would be associated with tolerance, again, in patients with severe disease. So it was encouraging that we found this drug that has been reported to work, although I should add that we haven't actually seen the data on how dexamethasone works in those patients yet. But that was certainly encouraging to see that we found this for MERS. I also think it's noteworthy that we found beta estradiol, and I'll talk about that in a moment. So I looked at some of the data that's already out there in terms of the transcriptomic data for SARS coronavirus 2 and just compared it at a very preliminary level with the MERS data that we have. And so I should add, this is extremely preliminary and more work needs to be done to make sure that this data is normalized in the same way as our transcriptomic data. But it's worth noting that we also identified dexamethasone in this again. We identified tocilizumab, which is an IL-6 inhibitor that is currently under investigation in clinical trials for COVID-19. And again, we identified beta estradiol. And I keep coming back to the beta estradiol because we have observed clear sex biases in SARS coronavirus 2 outcome. So this study was done with SARS Classic by Stanley Perlman's group in 2017. They found that there were sex biases that appear to be biological and not related to behavior or any type of gendered cultural differences, which has often been attributed to sex biases that have been observed in human patient populations. And here we show, or Stanley Perlman's group showed that male mice were more susceptible to death from infection with SARS Classic. They had higher levels of inflammatory macrophages in their lungs, and those macrophages were expressing pro-inflammatory cytokines such as TNF-alpha, IL-6, and IL-1 beta. And that furthermore, when they either overectomized these mice or treated them with an estrogen receptor antagonist, pardon me, I have a tickle in my throat, that estrogen receptor signaling was actually protective. And this is consistent with data that Sabra Klein's lab, in particular at Johns Hopkins, has found for influenza, and that estrogen and sex hormones in general, but particularly estrogen, is important for signaling and associated with protective immune responses, as opposed to some of these more harmful responses. And Akiko Iwasaki's group at Yale just released this preprint that showed that, in fact, there is a sex-biased immune response to SARS coronavirus 2 in patients. So they showed that both male patients have higher viral loads in their nasopharyngeal swabs and saliva compared to female patients. Female patients had higher levels of anti-spike antibodies in their blood compared to male patients. And male patients in general had higher levels of a number of different inflammatory cytokines compared to female patients. So there are clearly sex bias differences in both inflammatory cytokines and chemokines. And I think this is really relevant in terms of thinking about how we might treat male patients versus female patients, and some things to consider in terms of repurposing drugs. And this brings me into my plans to investigate this. So I've already been looking at sex bias in Ebola virus disease using the collaborative cross model that I spoke about a little bit earlier. And I didn't show any of that data today, but the bottom line is that we are able to see very clear sex biases in Ebola virus infection using this model in certain genetic backgrounds. So the collaborative cross, as I mentioned before, is this panel of genetically diverse mice They're derived from 8 different mouse strains, 5 of them conventional laboratory strains, and 3 of them are wild-derived. They represent about 90% of genetic diversity across the entire mus musculus species, compared to most conventional laboratory mice, which are only about 11 to 15% of that potential genetic diversity. It also covers the 3 main subspecies of mus musculus. That's domesticus, musculus, and castaneus. And these mice were generated by this funnel breeding scheme. Essentially, they took all 8 of these founder lines, bred them together in different configurations over 2 generations, and then started back crossing them, so that the result of each of these funnel breeding schemes is a different collaborative cross line with a different mosaic across from the 8 founders across their genomes. And they are 90 to 99% homozygous, so they're reproducible and inbred. And if you cross 2 of these lines together or cross them with another type of mouse, then those will have high heterozygosity. 2 CC lines crossed together have heterozygosity that's consistent with or even greater than human populations. So it's a great reproducible model of genetic diversity, which is very relevant to trying to understand host responses and how those occur in a genetically diverse population like the human population. And as I mentioned before too, we've seen this spectrum of Ebola virus disease phenotypes in the collaborative cross, both in pure CC lines as well as these F1 CC RIX crosses. We see about 1/3 of the mice are tolerant to infection, 1/3 of them are lethal, but with no evidence of hemorrhagic disease, and 1/3 of them are lethal with evidence of hemorrhagic disease. So this allows us to look at a much broader range of Ebola virus phenotypes than what we've typically seen in conventional laboratory mice, which is that they are lethal, but there are many of the features of the hemorrhagic disease, and the tolerant features are missing. So we wondered if we can apply this to COVID and use this model to investigate some of these sex differences. So work has been done to model SARS coronavirus 2 in the collaborative, in mice, but they can't use mouse ACE2 as a receptor. So there is a human ACE2 transgenic mouse that's a knock-in. So it has human ACE2 basically where the mouse ACE2 should be in the genome. And they can be infected, but they don't develop severe disease. So that's a problem, but they are in a conventional, I believe, C57 Black 6 background. So if we looked at this in a model that had increased genetic diversity, we might be able to see some of these expanded phenotypes. And furthermore, relevant to the issue of sex bias is that ACE2 is located on the X chromosome. So it's also possible to look for any type of genetically sex-linked effects that the receptor itself might have on virus infection in the context of a diverse genetic background. And David Threadgill, my collaborator at Texas A&M, who generates all of the CC mice that we use in this study, has already shown ways that he can find sex biases and phenotypic differences using a type of cross that is an F1 transgenic mouse crossed with the collaborative cross. And so basically what we would do here is take a transgenic female mouse that is homozygous for human ACE2, cross that with male CC mice, and then you would have offspring that are both positive for ACE2, as well as have the genetic diversity from their fathers, their sires, that would allow us to look at this increased range of COVID-19 phenotypes. So hopefully in this model, we could look at lethal disease and COVID pneumonia. We could look at the gastrointestinal disease and the reports that there may be virus in the stool and it may cause diarrhea. We could look at the different vascular or cardiac implications that have been observed, as well as the neurological phenotypes that people have also reported in some COVID-19 patients. So this is on the horizon. Also, I've heard that there are reports now that people have developed mouse-adapted SARS coronavirus 2 strains. So that's another option where we would not necessarily even need to have the transgenic mouse. We could infect collaborative cross mice with mouse-adapted SARS coronavirus 2 directly. So these experiments are ongoing, and check back with me next year, and hopefully we'll have some preliminary data from this. So now I'm just going to talk briefly about what's next, and then we'll get into the discussion period. So as I mentioned, we are right now in the process of analyzing this gene expression data from SARS-2 infected rhesus macaques, looking at host responses, looking at sex bias. Even though it's a small group of animals, there was a balance between male and female. So we will see if we can identify sex-specific host response signatures and start to perform some of these classification efforts, just distinguishing infected from non-infected animals, since all of these animals have the same outcome. To develop this mouse model of SARS coronavirus 2, as I mentioned, and then to integrate all the data coming from these models with our MERS, sorry, that should say MERS coronavirus, not MERS coronavirus 2, but with our MERS coronavirus data, and then further integrate that with data from public data sets across other coronaviruses, other animal species, and other species of viruses, and also include, hopefully, more human patient samples. And then develop and test these pan-species, pan-coronavirus classifiers, perform this integrated drug repurposing, and start testing some of these repurposing candidates in animal models of SARS coronavirus 2. And with that, I'd just like to acknowledge my collaborators, in particular, my collaborators at Rocky Mountain Labs, Atsu, Nilcha, Vincent, and Hines, and David Threadgill, who has provided all of my collaborative cross mice for me. And finally, acknowledge my funders. A lot of this work was supported by DARPA. Some of it was supported as well by DITRA and NIAID. And then we have a FAST grant to perform the SARS coronavirus 2 work that I was describing. And with that, let's get into the discussion period.
Thank you, Angela. That was fantastic. And thank you for sharing the preliminary data and work in progress with us. And among other things, I'll really look forward for the comparative gene expression data set. Speaking of gene expression data, let me ask you this. You've employed RNA-seq for your analysis. Have you considered DNA sequencing approaches? And a whole genome sequencing can be prohibitive in terms of cost and other logistics, but in whole exome sequencing, because I feel that would give you information about, say, SNPs. And because when you talk about genetic susceptibility, you can't help not thinking about SNPs. So what would your thoughts be on that?
Of course. So we haven't thought about doing that for any of the non-human primate studies, mostly because the group sizes are very small. So it's very difficult to do without even getting into all the issues that can occur doing like a genome-wide association study. But with the collaborative cross, we actually don't really need to do the sequencing because those animals all have been genotyped very extensively, and their genomes are all fully sequenced. The animals themselves are genotyped with a large genotyping array. So we can identify SNPs or even larger genetic loci that would be associated with these phenotypes. And one experiment that we have ongoing now for our Ebola work is that we've screened enough different collaborative cross genetic backgrounds that we can actually begin to do some of those quantitative trait loci analyses, looking for the regions of the genome that are statistically significantly linked to outcome, and begin to compare those regions of the genome once we have them mapped to our transcriptomic data to see which genes within those QTLs might be actually contributing to the phenotypes. So that work is ongoing, but we don't have any data to share yet.
I think I'll jump in. I think Sarada was asking about human gDNA, like from a cheek swab or something like that.
Right. So we haven't done anything like that. And actually that is definitely not my area of expertise. But I do think that other people are doing that. So it will be very interesting to see, especially once they get the type of numbers that you would need to really strongly associate some of those SNPs with outcome or phenotype. So certainly that work's being done, although I'm not the one doing it.
Right. Yeah, we've got Jean-Laurent Casanova coming up in about a month or so. So hopefully he'll fill us in on that.
Yeah, I was going to say he'd be a great one to talk to. And I know that at the New York Genome Center, they are doing that type of work. And I've certainly talked to people who are interested in getting access to large patient cohorts where you could really get those kind of numbers to figure out what, the genetic underpinnings are in the human population.
Yeah. Let's see. I've got some questions here. I found it interesting that the human ACE2 knock-in mouse, you say, don't get serious disease with was that SARS-1 or SARS-2? I missed that.
That was SARS-2.
Oh, okay. But the human DPP4, that's made the same way of allelic replacement, and they do get serious MERS. Is that?
Yeah. So they do get serious MERS. There is one thing that is a problem with some of the DPP4. There are several transgenic mice. Some of them are knock-ins, some of them are not. But many of them also develop encephalitis and these neurological disease. They all, to my knowledge, have replication that occurs in the brain. And in our model, I didn't show the data. There is virus replication in the brain. There's just no evidence of neurological disease. So we've continued to use this model, but as with many different transgenic models, that is one of the drawbacks to using it, because as far as we know anyways, neurological involvement with MERS coronavirus in people is not really is not thought to be a major feature of that disease. So that is one of the drawbacks to this. To my knowledge, there hasn't been reports of the same thing happening with the ACE2 mice, but I'd have to actually go back. There have been some more preprints out about different mouse models. There's also another approach being used by Stan Perlman's group to sort of transiently express human ACE2 using adenovirus vectors to deliver ACE2 to the respiratory tract. So all of these models have their, benefits and drawbacks. I think, we still need to do a little bit more to look at the ACE2 transgenic mouse. And
yeah.
I think that also it's definitely worth exploring some of the mouse adapted models that don't require a transgene at all.
So, sorry, I missed that. Do you think that part of the problems with the model is the proposition that there's some loss of function of the ACE2 or the DPP4 in its endogenous role from the grafting of the human mutations, or no? Sorry.
I don't know that. What I think in the human ACE2 transgenic mice for SARS-2, I think the problem might be what we've seen in a lot of other mouse models in general, and that is that you end up only seeing the phenotype that particular strain is allowing for. And certainly that's what we've seen with Ebola, where if you infect a C57 Black 6 or a BALB/C mouse with Ebola, they'll die. The virus targets the same organs that it normally targets in people, so the liver and spleen, but there's no evidence of hemorrhagic disease. And the tolerance in that is usually dose dependent. Whereas if you look in a variety of different strains, you will see an expansion of the phenotypes because really of the entirety of the rest of the genome and its genomic diversity compared to just the one, single BALB/C mouse.
Yeah. I've got a lot of stuff about the mouse strains. I'll circle back to that. But since you mentioned with the MERS, the neurological symptoms, I wanna ask you, what's your take on neuro COVID and the all the vascular symptoms, etc. And I'll give you a little background about why I'm asking you this. We had Florian on last week, and Florian Kramer,
Mm-hmm.
and
yeah.
I asked him, and he said, he made some statement along the lines of, and SARS-2 is a classical upper respiratory infection, period. And I said, Loren, that's not really my understanding. I thought we were getting, all these pulmonary embolisms, neurological symptoms. And he said, oh, you're gonna see that to some extent when, if any pathogen, if infects many millions of people, you'll just get these tails that you don't see in normal infections. And he kind of, basically said there's nothing to see there. What's your take on, are those real? Should we be worrying about those things, or are those just long tails?
Well, I sort of agree, and I also don't. So I think that, but then again, that's really thinking, that's a way of answering it that really focuses on the virus. And I focus on the host. And so I certainly do agree with the point that we would see, you always see a more diverse spectrum of outcomes with almost any viral infection, the more individuals that you put it into, because people have different host responses. Even when you look at like the collaborative cross, even if you look at the controls and you compare them to each other, they have very different baseline levels of transcriptomic just gene expression across the entire transcriptome. So even by themselves, without any infection, they're different in terms of just what they're expressing at any given time. So you would expect that diverse group of individuals, and especially in a more diverse population, large population like humans, you would expect to see people with different responses. But that said, I don't think we know enough about COVID or SARS coronavirus 2 infecting other tissues. I think that's one thing that we really don't know very much about at all. And it's clear that it probably can infect other types of tissues, but it's not clear, for example, the gastrointestinal manifestations of this. So not very many people have diarrhea, but now we've seen all these reports that a lot of people, but not all, again, have virus in their stool. It's not clear to me that they're getting that in their stool from ingesting the virus, because I don't see how an enveloped virus could go through the stomach. Yeah. So it's getting in there some other way. And it probably is infecting those intestinal cells because they do express ACE2. They probably have furin or other proteases that can process the protein and enable fusion. So I don't think that we can completely say that it's just in the respiratory tract and this is some weird, tails that we're seeing, but they're not typical. I mean, they may not be typical, but that doesn't mean that they're not caused by this infection. And I would argue that some of the features, such as the vascular issues with this virus, are very consistent with other viruses that cause systemic inflammation, such as Ebola. And even though COVID-19 doesn't look like Ebola virus disease, it's thought that the major mechanism of coagulopathy and vascular leakage in Ebola is actually due to these systemic cytokine storms occurring. It's not because the virus itself is infecting the vascular endothelium, even though it actually can do that. But they've shown in studies that it's not like virally infected cells dying in the vascular endothelium that are causing the activation of these vascular surfaces and this uncontrolled coagulation and vascular leakage. It's actually those pro-inflammatory cytokines that are being dumped out by infected macrophages. So there's a lot that we don't understand about this virus, including which cells it infects and the effects that the systemic effects of these inflammatory cytokine storms that are being created. It's totally plausible to me that you could have the bulk of the infection occurring in the respiratory tract, in the lungs, but you're still seeing these weird, embolisms and blood clotting disorders because these cytokines are causing that effect to occur. Right. So I think that there's still just a lot more that we need to understand, both about viral tropism and about the sort of larger systemic effects that these host responses in infected tissues are having.
Okay. Now I want to circle back to the mouse model. Obviously we all love the mouse model, and I particularly, I do a lot of GWAS in my spare time. So I love the collaborative cross, but it seems like it's a little problematic for immune stuff, because just, of the fairly profound differences between the mouse and human immune systems, the inversion of the role of monocytes and neutrophils, the swapping of interferon gamma with interferon alpha beta. They don't express MHC on their T cells. So I guess I have multiple questions here. I mean, one, it's amazing that, you guys still got, 75% predictability on from mapping from mouse to human. And I guess, so a sub question would be, you said you mapped to the orthologs. Do you think you would do better if you, started obviously by mapping to the orthologs, but then in places where like roles were known to have swapped, if you would then go and manually edit this to, the thing that it's known to do instead?
So that's certainly one way that we could refine our predictive capabilities, because that is a very good point. And often, just looking at trying to develop those signatures based on expression alone has led to some problems, because we end up identifying something that has no ortholog if we start with one species, or there's, this problem gets compounded when talking to mathematicians about this, because they're like, wait a second, like you just said that this is similar to that one, but this annotation database is telling me that there's 5 other genes that are called this. And so it can be, that is a real challenge. So what we did with the Ebola stuff, just to kind of verify our techniques, was we actually did it both ways. So we used the same approach to identify potential predictors in the human data, and then we mapped those back to the murine orthologs and tested it in the mouse data set. And I didn't show that, but using the human data to test for outcome in mice, we got 90% prediction accuracy. So it was a little better going from human to mice than it was from mice to humans. Okay. But that's why we thought it actually might be better to include other species as well. Yeah. And one of the reasons for that, the non-human primate models are very good in terms, they're considered the gold standard for modeling filovirus pathogenesis. But they actually can be also more diverse than humans. And even in a large study that we had, which was 6 mice or 6 monkeys per group, right, which is a lot of mice for a BSL-4 study, or monkeys for a BSL-4 study, that's still not very many individuals. It's hard, but by looking for orthologs. What's that?
It's that it's hard to power a GWAS with n equals 12.
Yeah, it's just not going to happen. And that study, like I said, was pretty good, because we actually had 10 groups of 6 animals, so it's 60 total animals, which is great. But we thought that we would actually be able to find orthologs that would be more reliable if we incorporated more species into it. Mm-hmm. So then we're not even worrying about that gap, that functional gap that you pointed out between mice and humans. We'd be able to have some other species in there. And especially as we try to expand this to include other viruses, then we might be able to keep incorporating other species in there as well, and hopefully just refine orthologs that are more robust predictors across all these different species.
Yeah, that's excellent. Okay, next up we have a question from Charles. So we've asked most of our speakers so far this question. You're probably better positioned than anyone so far to address it. Basically, what's the deal with ACE2 and DPP4? Why do the viruses, like to target these cell surface proteins, that they've got, tens of thousands to choose from. So from the host side, do you think that there's anything special that led these to be selected?
So I don't know, and I may not be the person to answer this question, because I think that this question really becomes an evolutionary question.
Absolutely.
So these viruses, we think that they mostly come from bats. And bats themselves, I mean, I've been working with my colleague at Columbia, Simon Anthony, who studies virus ecology, talking about bat receptors. The same is true actually for Ebola, but at least with Ebola, the receptor NPC1 is highly conserved through, all vertebrates. It's a cholesterol transporter, so it makes it a little bit easier. But bats themselves are so diverse that and we actually don't know very much about many of their genomes, so it's really hard to make comparisons. And then when you get into the structure of these things and how they might interact not only with spike from either MERS or SARS or SARS-2, they might be completely different for the bat coronaviruses that led to these viruses, as well as the other bat coronaviruses that might be circulating in the same bats and might be recombining with them. So it's a really complicated question to ask, and I think a lot of it is really this evolutionary component that, humans aren't getting these viruses until there's something that disrupts their normal ecological niche that they exist in these different bat communities. And we just don't know a lot about what that process looks like. In some cases, there may be an intermediate species, and we certainly don't know if there is one for this SARS coronavirus. I mean, there's certainly that, intriguing finding from the pangolins that the receptor binding domain of the pangolin coronaviruses is very similar to this one, which otherwise looks very bat-like. Right. But ultimately, we still don't really have that. We haven't found the smoking pangolin or the smoking bat even. So we really don't know how all of those processes occurred. I think, though, from a host response standpoint, I mean, it's certainly interesting in particular for ACE2, and I think it may, in part, explain some of the COVID-19 disease features that we're seeing, and that ACE2 has been targeted for hypertension, as this question says, because it's an important regulator of blood pressure and vascular permeability. And it's completely feasible to me that virus binding ACE2 may have some sort of off-target effects by inducing signaling through that receptor that's having these downstream consequences in terms of the pathogenesis. But why it occurs in one patient versus another, that's an excellent question. And I know that so far, nobody's found an association between the different alleles of ACE2 that are known. But I think that story is by no means complete. I think that a lot more work needs to be done to sort of understand the role of ACE2 in mediating host responses, just as it needs to be understood in mediated viral entry.
Absolutely. Yeah, I wouldn't say that there are no reports. I would say there are no ironclad reports. I've seen a couple smallish studies. One was on just the coding SNPs of ACE2, and I think they said, oh, it looks like, these three might have some association. And then the Italians, since they got a head start on this whole thing, they had a very small GWAS that suggested that there were some non-coding SNPs of ACE2 that seemed to regulate expression level that might have some correlation.
Yeah, and that makes sense. I mean, certainly, there have been a lot of studies about expression level of ACE2 and a lot of assumptions made about it. But the truth is, and I mean, this is what is often hard to communicate to the public too, is that we can do all of these studies and they're all pieces of information, but we still haven't really put a lot of them together. So just because there's no evidence of something doesn't mean that it's not happening, but in many cases, we still just don't know the full picture because we're still at the stage where we're collecting all these different little pieces of information, and we need to be able to put it all together. I'm kind of impressed, though, with like the job that science as a whole, as an enterprise, has done, considering this virus we didn't know about before 6 months ago. There's now, thousands of preprints and studies about it. And frankly, it's hard for me to keep up anyways with everything that's been published. But that's another reason why seminars like this are great. It gives you the opportunity to kind of get other people's thoughts on all of these little pieces of data, and it's helpful to me anyways for trying to see the bigger picture.
Mm-hmm. Sarada, I've got one more. Did you want to pop in with one, or?
Yes. So I have a question. So this is referring to your comparative gene expression data, and I believe you looked at the lungs. Have you looked at other organs? And if so, do you see any difference?
For MERS, do you mean?
Mm-hmm.
Yes, so we have actually looked at nasal swabs and at PBMCs. And to be honest, I'm still in the process of analyzing that data. The nasal swabs have been very challenging because we've been looking, so that's a comparison of camel, alpaca, and marmoset nasal swabs. And the camels and alpacas have been a problem and a bioinformatics challenge. So one thing I didn't mention in this part of the talk is that we are engaged with Michael Kirby, the mathematician that I collaborate with, to do a more comprehensive ortholog map between the camels and the alpacas. And we're ortholog mapping them to humans, which is what we did before, but based on sequence rather than on annotation. And the problem with the annotation-based mapping is that both the camel and alpaca genomes are sequenced and they are assembled, but those assemblies are little more than like a bunch of contigs.
Yeah.
And so, yeah, I mean, we have all these features, like these differentially regulated transcripts, but the majority of them we can't even identify. And in terms of, and that's why we haven't actually dug so deep into those studies, because we know that we are missing probably a lot of information just using the annotations for the orthologs. So the problem is with the sequence-based is that it essentially means blasting everything in our transcriptomes against the human genome. Because we've decided to just save ourselves the trouble of trying to blast it against everything and just normalize everything to the human reference genome. And it's just very computationally intensive. And since there are other computational demands and we only have so much processing power, it's slow going. But our hope is that once we do have those improved orthology maps, we'll be able to look at those samples much more closely and also be able to compare them with human samples and non-primate samples that we do have those same tissues for, as well as better data.
Oh, yeah, genome assembly is something I guess Loren can easily relate to. Okay, here's one other question is probably a general comment from probably a very naive comment. My understanding was that the mouse and human immune systems have some very basic differences. And it's pretty impressive that in spite of these differences, you're still able to use predictors from mouse data to use signatures of mouse gene expression as predictors for human disease susceptibility or even drug responses.
So one of the things about that is that, yes, there are these differences. And, there was a paper in 2013 from Shaw Warren's group that showed that there was like no correlation at all between transcriptomes from people who had a variety of different inflammatory or traumatic injuries and mouse models of the same thing, which was pretty depressing. There was a New York Times article about it at the time that was like, are mouse models like even worth it? And I was like, oh God, this is terrible for somebody who works with a lot of mouse models. But one thing that we have seen is that similar to the improved fidelity of the phenotypes the disease phenotypes that we see, at least for Ebola, but this has also been shown by other groups working with flu and SARS and West Nile virus in the collaborative cross, that genetic diversity does improve that somewhat. So you're not just looking at, essentially one individual, which is what an inbred mouse line is most of the time. Most humans are probably not going to be very similar to a genetically not very diverse mouse that's the result of like 25 generations of brother-sister pairing. It's just not going to be very comparable, but when you add that diversity in there, it becomes more human-like. So even though individual functions might not be exactly the same or exactly comparable, when you're looking at it over this more diverse, you will see some of the same pathways at least. And in our case for the predictors, we actually don't necessarily restrict it to just immune genes. It's any type of gene that might be differentially regulated in the host response. So in viral infections, usually things that are differentially regulated include things like metabolic genes, things that are involved with stress responses, cell cycle control, and sort of basic cellular functions, because viruses a lot of times will appropriate these basic cellular pathways that the cells just, translation and endoplasmic reticulum organization and trafficking and things like that. So a lot of these pathways or these genes that are in our classifiers might not necessarily be related to the immune response. The other thing is some of them are, but some of those are more conserved. So there are things like, certain inflammatory genes or interferon stimulated genes that are common to both human and mice. So even though there's, differences in the way that monocytes function in mice versus humans, there might be differences in T cell signaling and the things that they express. The genes that we've chosen are not necessarily affiliated with that. And sometimes if we, because we are using this sort of iterative approach to get out individual genes in these classification signatures that don't work very well, some of those things that don't necessarily apply to humans or mice or vice versa, we can filter out during that stage so that we have a classifier that can work robustly across both models.
Oh, we have another question from the audience. Louise would like to know, where is the best alpaca genome contact information?
Good question. We are using the one on NCBI. I'm not sure there's an alternative. I wouldn't say that it's the best in terms of the best you could do, but it may be the best that is accessible. I do need to look and see if recently any new updates to that have come out, or if there's like an ensemble genome or something. But I think for now, the only genome I'm aware of, at least that we were using, was the NCBI one. So there is a dromedary camel genome project, as well as a Bactrian camel genome project. Perhaps that may have come out with some better annotation, but I'm not sure. I'm sorry, Louise.
Yeah. Hey, I've got a perspective question about something you alluded to. So, you've shown that the omics can be remarkably predictive in your mouse models. And, we're going to assume that omics will be super predictive in humans as well. What sort, so I guess RNA-seq, but on, what sort of tissue, time frame, et cetera, would you think? And, how many knobs that the doctors can turn do you think we have at this point?
That's an excellent question. I think that ideally, the best tissue you would want to use is like something that would be easy to collect. So for Ebola, we started in the mice. Actually, I should add that prediction is even better because it was across tissues. We used spleen data to generate the signature in mice, and then we used whole blood data that was essentially left over from diagnostic PCR in the human cohort. For SARS-2, I would say that probably either nasal swabs or blood would be the gold standard for something that you wanted to turn into like an actual diagnostic or prognostic assay. Yeah. In terms of though the knobs that physicians could turn, that's definitely a work in progress. I mean, even, at the beginning when I was kind of talking about antivirals versus modulating host responses to induce tolerance, we've actually seen some examples of that in this pandemic. So remdesivir, which also failed trials for Ebola because it didn't really work very well, but it was tested in patients who were, showing up at the Ebola treatment units or the ETUs once they were already sick and symptomatic. And at that point, it may be that the ship has sailed and targeting the virus won't help. But if you are able to identify a patient early with SARS-2, it's possible that you could treat them with something like remdesivir, especially if they're pre-symptomatic. I mean, if you could control the infection before they started, down the path of these potentially harmful host responses, that might be one thing that a clinician could do to improve that patient's outcome. And sort of conversely, if you had a patient who is already symptomatic and, our predictive data says they're going to get really, really sick, I know that there are trials going on right now of these different immunomodulatory drugs, which we may be able to apply some of that information, whether it's, tocilizumab or, dexamethasone once we finally see the data. It's totally possible that different outcomes at different time points in infection or the clinical course of disease might result in different clinical guidance and recommendations. But right now, I mean, I think all types of countermeasures are really very much a work in progress in terms of what really is available to treat patients, especially those that are looking like they're, headed to the ICU.
Yeah, I mean, I on my list of potential wonder drugs, I think my number one is actually tocilizumab. So it would be great if we had, and you must know this, so that's IL-6 receptor. Do you get, does one get bifurcations in hyperimmune interactions where some can go an IL-6 mediated pathway and some can go a different way? So there might be another tocilizumab out there?
Yeah, so that's an excellent question, and it's one that we don't really know. I mean, most of the data on these cytokine storms are different group sizes of patients. So I think on one hand, we're going to have to wait for like a big meta-analysis of a lot of these different studies to try to figure out if there's like one pro-inflammatory cytokine or multiple that are driving this. I know that in addition to tocilizumab, I think there's an IL-1 and a TNF-alpha inhibitor that are also being tested for this type of, cytokine storm modulation. And it may be that, in some patients, there's a combination of them. It's totally plausible, I think, that there could be multiple pathways, like two cytokine storms. And we know from the data that Ben Tenover published, his transcriptomic studies looking in patients in mice, in ferrets and in primary cells, that it does look like in all of them, just infection induces lower interferon innate antiviral responses than we would expect. And because those antiviral responses are so tied into the induction and regulation of inflammatory signaling, it could really go in any way. So we really just need, we definitely need a lot more host response data from patients as well as from animal models. We also need to be able to sort of consolidate all of these different like sort of observational cytokine studies that have been associated with different patient outcomes and try to figure out if we're really dealing with multiple types of cytokine storms, or if, you know, there's going to be like one inflammatory antagonist to rule them all.
Yeah.
I think the jury's sort of still out on that, and we just need more information.
Yeah. One theme we had been maybe 3 weeks ago. One theme that emerged from his talk was that, you know, the innate response is probably what casts the die for you. You know, the interferons, but you know, I worry that, you know, since that's probably going to be so early that you're not going to be at the doctor at that time. You know, that you know, we are going to probably have to play catch up with cytokines and adaptive responses and et cetera.
Absolutely. And I think, you know, as we are better at detecting, you know, this early, I mean, a lot of this is stuff that we do sort of know how to do, but we can't do it for reasons that are pretty non-scientific. Like, we need to test everybody like routinely, especially in places that have an upswing of cases, and we're still not really able to do that sort of basic testing as well as the follow-up epidemiology that you know, we also know works, like contact tracing. So maybe we could find people who are pre-symptomatic where that die really hasn't been cast yet. But right now, I don't think that we are anywhere near being able to monitor people who are at risk of infection and catch them early. From my understanding, people still have to sort of seek out testing, and there's still a lot of mixed messaging in terms of, you know, I've talked to a lot of people who don't seem to understand that just because you tested negative today doesn't mean that you're going to test negative tomorrow. And because we just don't have the capabilities to do any of that, and there doesn't really seem to be strong messaging going out that that's kind of the direction we should be going in, I think it's going to be a real challenge to try to catch people as early as possible. So
Yeah.
we may just be, you know, ending up having to make recommendations and develop classifiers and interventions that deal with the patients that we have and not the ones that, you know, we would want to have.
Right. Or if we could, you know, if we can come up with a prophylactic, you know, before a vaccine, if it's like, oh, this drug will really ramp down your response and make your outcome more favorable.
I mean, even better. Yeah, I think that something like that would be incredibly helpful. I certainly hope that if we are able to find something like that, and I hate to even say it, it's something like hydroxychloroquine that's cheap and widely available, and not something like tocilizumab, which is just
Right.
not feasible for us to roll out as a prophylactic at population scale. So, you know, I certainly hope that we find something like that. And, you know, I should just say I've never seen any indication that hydroxychloroquine works as a prophylactic. So
Right.
don't take that as me endorsing it. Certainly don't start taking dexamethasone as a prophylactic.
Yeah.
Probably is not a very good prophylactic, and could actually make people more susceptible to severe disease later on. But something like those drugs, something that is in every pharmacy, that is manufactured at scale already, and that is cheap and easy to access, I think would be ideal. And hopefully, you know, with all of these different efforts to, I mean, I'm certainly not the only one who's come up with the idea that maybe we can use drugs that we already have. And there are other people using different approaches to identify candidates for repurposing. Hopefully one of these strategies will result in identifying something that could make a measurable difference in terms of prevention.
Yeah, I mean, so the one hopeful thing that I've seen data for, we had it in our series, was from Pepsid. Have you seen that data? And what do you think?
I've heard people talking about it. I need to look at it more closely. But yeah, I've heard that famotidine which is really good because I have heartburn a lot and I take Pepcid. It's Pepcid, the stuff that's like combined with Tums is like my total jam, like when I'm eating pizza or something. But I need to look at that more closely. I mean, the trouble with the drug repurposing stuff is that there are actually so many different preprints and reports of either something worked or it's predicted to work, or it works in vitro, or maybe it works, but here's a very tiny observational study. And it's really hard to sort through just the mountain of stuff and try to make a conclusion about anything until it's been through a clinical trial that obviously is just hard to do, because in any situation like this, you know, the priority has to be patient care and not conducting like a perfectly designed randomized clinical trial. So, but I will look into that more. It's hard to keep track of like what's the most promising today.
Yeah. Hey, we should probably let you go. I just wanted to ask you one more thing. What do you think are the top couple unknowns that aren't being looked at enough? And if you could, point to a department and say, if you guys could focus on these handful of things, that would be huge.
Well, I think that one of the biggest unknowns that is not, I mean, I'm a virologist, so it is within my wheelhouse sort of, but it's very hard to figure out, but it's also incredibly important. It has to do with transmission, which I don't really study. Mm-hmm. And that is what is the minimum infectious dose for people? So what we've seen in other infections, other models, is that there, for the same virus, there may be drastically different minimum infectious doses required, whether you're getting the virus through your nose, whether you're sort of inhaling orally respiratory droplets versus conjunctival exposure, things like that. We don't really know anything about that. And it makes it very difficult to determine risk. Mm-hmm. It also makes it very hard to understand how host responses can affect transmission. And I know Sarada mentioned that you guys are having Akiko Iwasaki to come talk with you. I'm sure about her exciting sex bias paper, but also she had a paper out a couple months ago that I thought was just really well done and really fascinating. And it showed that for influenza, humidity actually not only made a difference with the stability of the virus, but it made a big difference in terms of the host response. And it determined whether the animals, it determined their susceptibility. Mm-hmm. So I think that looking at minimum infectious dose in different circumstances and by different routes is hugely important and really needs to be understood, so that, especially, it looks like we're in this for the long haul. That will make it much better for people to make public health recommendations about how to reduce transmission in really, practical, actionable ways. Mm-hmm. And I think the other thing is that we just really don't know very much at all about host responses to this virus. Most of the work that's done, focuses on the immune response, which is still incredibly important. And it's really important to have these easy laboratory indicators of outcome, correlates of protection. People are looking at now D-dimers and C-reactive protein and all these things to try to figure that stuff out. But we really just need more like global host response data. And whether that's transcriptomic or proteomic or whatever, we really need that larger scale data. We don't all need to be looking at the same exact panel of 12 cytokines or whatever. We really need to have that data from a large cohort of patients so that we can start looking not only for host targets for treatment or for diagnosis, but so that we can really start to understand pathogenesis. And that's going to be really critical for trying to make sense of all these different phenotypes that are occurring. I mean, right now, we don't even know, all of these tissues that are infected. So we certainly also need more data like that, just on where's the virus going? If we could, even look at that with autopsies, and also now that more and more animal model data is becoming available, we'll be able to use that to start start answering some of these questions so that maybe we can ask we can make more informed hypotheses about what's going on in people going forward. But I'd say that those are two of the biggest things that are still really open questions. And I'll let Akiko certainly, and probably Florian brought this up last week, but there's obviously still a lot of questions to understand about protective immunity and long-term immunity too.
Yeah, so Sarada, I guess we've got a comment from Darcy.
Darcy, yes. So Darcy has a comment. Heard a very interesting talk by the head of Decode last week. Extremely interesting data from Iceland. Solid data on different age cohorts. Good genomic evolution data, not so much genetic diversity. I'm going to put this in the chat box for you.
Yeah, I haven't seen that study. Does that refer to genetic diversity in the hosts or for the virus?
I assume that means the host.
Yeah.
Pretty homogeneous population.
Yeah. I mean, I'll have to look at that. Like the data from Iceland has been very interesting in general, just because, well, I mean, frankly, their response and approach, and again, I'm not an epidemiologist either, but it seems like it's been pretty effective at controlling it. Then again, they are also an isolated island nation with a much smaller population than ours. But I will have to look at that. I'd be very interested to see some of this data. And I think that that's really relevant too in trying to just assess risk. We've made these assumptions, and it does appear that there is a severity bias that's associated with age. But it's, you know, that those age and medical status are sort of broad categories, and we still, I think, don't really understand how all of that works. So I'll definitely check that out. So thank you, Darcy.
Yeah, that was truly amazing.
And see, I didn't lie when I said we love to talk.
I love to talk too, so it's no problem for me.
Yeah, well, thank you so much
for your time, and thank you for staying back for this super interesting, but rather long discussion, but we really enjoyed it.
Oh, it's my pleasure, and I'm going to try to be in attendance at more of these seminars coming up. This is, you know, honestly, the last few months have been surreal, I think, for all of us, but it feels great and sort of like a return to normalcy to be giving a scientific seminar and having, you know, great conversation with colleagues. So I didn't realize how much I actually missed doing this. So it's been wonderful, and thank you so much for inviting me and having me here. And I'm looking forward to watching some of the future seminars in the series.
Excellent.
Same Zoom link every week.
Yep.
Excellent. Well, I'll keep an eye out. I follow you both on Twitter now, so I'll.
All right. Thank you. Take care.
Thank you. Take care. Bye-bye.
Bye.
Bye.